Enzymes. So enzymes are considered as a biocatalysts. So here they are synthesized inside the living organisms and they are having a specific property called as catalytic ability. That is, these are the protein molecules actually. These are the proteins with a catalytic property. Due to the power of specific activation or specific activity. So this is a property that is related to the biocatalyst called as enzymes, which was given by Dixon and Webb scientists. So actually here these enzymes are discovered inside the living organisms, and these are present inside the living organisms. At the same time, they are required for many biochemical or metabolic chemical reactions which are taking place inside the plant cells and also in the animal cells. So without these enzymes, no reaction will be taking place in a faster manner. That is, their property is fastening or making the reaction to be completed in a short period of time. All the enzymes are proteinaceous in their nature, but some exceptions are there, except some of the proteins, some of the enzymes, those are called as ribozymes. So ribozymes are also the enzymes, but these are not having the proteinaceous nature. So example like here the RNA will act as enzyme molecule. So this RNA which is acting as an enzyme molecule, it is not a proteinaceous molecule, but it is a nucleic acid. And the best example for this one is 23S rRNA is one of the example for ribozymes. But apart from these few exceptional cases, all other enzymes are proteins. And we can say that in proteins, different categories are there, but one of the category in the proteins is the enzymes. That's why we call all enzymes are proteins, but all proteins are not enzymes. And why these proteins are the enzymes which are proteinaceous in their nature, why they are so called means because of their catalytic ability. And here these are actually organic compounds or organic molecules, and these are having specific activity or catalytic ability. These are active at minute quantities. So this is one of the property related to the enzymes. And here there is a difference between the other catalytic compounds which are considered as inorganic compounds. So but many such differences are there between the organic catalytic compounds like enzymes and the inorganic catalytic compounds, but the major difference is, so these enzymes are generally become inactive, or we can say that destructed at high temperatures. But the inorganic catalytic compounds these are generally active at high temperatures. That is a basic difference that we find between the organic catalytic compounds, that is enzymes, to that of other, that is inorganic catalytic compounds. And apart from this one, here we can say that some of the living organisms which can survive at high temperatures, like in sulfur springs, so those organisms also have the enzymes, and enzymes which have been derived from those organisms are functional or active at high temperatures at about 80 to 90 degrees of centigrade also. So these are the exceptionals where we can call them as they have been derived from the thermophilic organisms. So these are some exceptional cases, but in all others the enzymes are active only at the optimum range of temperatures. And here this is a basic difference that we find between the organic catalytic compounds, that is enzymes, and the inorganic catalytic compounds. Other difference is these are active at high pressures also. So this is one other difference between the organic and inorganic catalytic compounds. And likewise here the organic catalytic compounds are produced inside the cells, and they are catalyzing the chemical reactions or the metabolic reactions which are taking place inside the plant cell and the animal cells. So let us see how the discovery related to these enzymes have been found. Let us see the discovery related to the enzymes. So there is a scientist, that is Louis Pasteur. While he was studying the process of fermentation. So as we know that fermentation is the process of production of alcohol. So this was being studying in an unicellular fungi called as yeast. So there he come to know that he had noticed that some force or some vital force is responsible for the process called as fermentation, and he called this vital force which is responsible for fermentation as ferment. So this is the first discovery related to the presence of enzymes in the living organisms. And later W. Kuhne had given the term enzymes. N means inside, zyma means yeast. So as they have been first identified or discovered in the yeast, so the name was also derived based on that concept only. Then Edward Buchner. So here Louis Pasteur had said that something is responsible for the process of fermentation in the conversion of in the formation of alcohol, but he haven't given the name for that compound, just he said it as a ferment. But Edward Buchner, he had said that the ferment which is responsible for the process of fermentation in the yeast is called as a zymase. So because it had been discovered for the first time in the yeast, that's why the name was also related to that concept only. So he called that ferment substance which is responsible for fermentation and the production of alcohol in the yeast is called as zymase. Next scientist is James Sumner. So he is the first person to extract the enzymes from the living cells and making them into crystal form, that is a crystallization of enzymes was first time done by James Sumner, scientist. So the enzyme which was crystallized by this scientist is urease enzyme. So this urease enzyme was extracted from jack bean. The scientific name is Canavelia sinensis. And he, after extracting this urease from the jack bean, he had converted them into crystal form, that is crystallized the enzyme. So urease is the first enzyme to be crystallized. And what is the significance of making them into crystallized form is, so it is the best way to study the actual nature and properties of the enzymes. And after making them into crystal form, he said that the enzymes are proteinaceous in their nature. Then after this, there are many such scientists who had studied about the characteristic features and the properties of the enzymes. In that John Northrop, So, he was a scientist who had written a treatise related to the proteinaceous properties related to the enzymes. Likewise, he not only studied about the proteinaceous nature of the urease, but also he has studied about the proteinaceous nature of various enzymes like pepsin, trypsin, etc. So, this is about the discovery and the history related to the discovery of the enzymes and their study by various groups of scientists.